So I need to take the screen, right?
I'm going to introduce you here first, and then
okay.
I'll pass it over to you, and then yes. All righty. Well, thanks everyone for joining the meeting today, again, virtually. Again, here's a list of everyone that's helping us out here, our officers that are helping out with the organization and really the running of the group. And I will point out, of course, now we have just as of a few moments ago, we have a new chair-elect, Ben Garcia out of the University of Pennsylvania. So he will be stepping into that role.
So Chris, are you showing, have you got an active screen? Because I'm not seeing anything. But...
Oh, yes, I am.
Is everyone else?
We are seeing it.
Oh, well, we won't worry about it. I don't have to.
Okay. All right. Hopefully everyone else is able to see my screen, and then Mike will, we can work on your slides here in a moment once we pass the ball over. Hopefully,
okay.
It will all work out. And then thanks again to all of our sponsors that we have that have been supporting us throughout the years. And without further ado, we have Professor Michael Gross here from the Washington University that will be giving a presentation today. He is a very distinguished professor of chemistry, medicine, and immunology, principal investigator, and he serves as a NIH mass spectrometry research resource at Washington University. Notably, he is the recipient of the 2020 ASMS John B. Fenn Award for Distinguished Contribution in Mass Spectrometry for innovative and integrative mass spec-based footprinting for structural proteomics. His research goals is in the development towards the development of biophysical methods that use mass spectrometry to understand proteins, their interfaces, solvent accessibility, affinities of binding, and their folding and unfolding. And the title of Mike's talk today is going to be High Order Protein Structure by Mass Spectrometry Methods, Structural Mass Spectrometry. And so with that, Mike, I will hand the ball here over to you. Are you able to see share the screen at the bottom icon?
Yeah, maybe I have to get on this one.
Harsha, as the host, do you have to define Mike as a host as well?
Professor Gross, you just have to go to the bottom of the screen where you see a panel, and it starts with security participants and so forth, and then in the middle of the panel you see share screen. So you go there and...
Yeah, I do, and I don't see that. Oh, I got it.
Yep. So you just hit on that and pick your screen that you want to share.
Right. So how are we doing?
We are looking at your screen.
Yep.
Okay. So are you seeing it in the presentation mode?
No, not yet.
So I pressed presentation mode. Maybe I have to go back and share the screen again.
No, you don't. Do you have two screens? If it's on dual screens, you might be seeing it on the other screen.
Yeah, I'm seeing the full presentation. So when I went to present mode, it switched to my other monitor.
Yeah, I think you have to use duplicate, right? Duplicate the two monitors so that we see your presentation.
How's that?
We still don't see the presenter mode. We see the slide deck.
You may have to try to unshare and then reshare the screen. perhaps?
No, I think it's something to do with duplicate screens, right? So you probably need to show the extra screen.
So I have new share. Maybe I hit the wrong one.
Yeah, now we see.
Now we see it. Looks perfect.
Perfect. Yep.
Okay, great. Sorry for them. I should know better, but I stumble through this all the time. So first of all, I want to thank Delaware for changing our national nightmare. I hope that doesn't hurt anybody's feelings and giving us Joe Biden. It's been a happy week for me, at least, and for a lot of people, of course. I guess an unhappy week for a lot of people too, but anyway, I told my students I was going to go to Delaware and give a talk. I guess I'm not in Delaware, though, am I? I'm in the Delaware Valley. So I was a postdoc at Penn many, many years ago. And then I was a postdoc with Fred McLafferty. And I started at the University of Nebraska, just as a brief introduction. And at Nebraska, I organized National Science Foundation mass spectrometry resource. And we worked on ion chemistry and instrumentation and environmental chemistry. Hard to believe how our subject has changed in 50 years. So I've been at it for over 50 years. And when I came to Washington University 26 years ago, I came here because I wanted to become more active in biologically related mass spectrometry research. You know, and it was just the beginning of electrospray, right? And what a revolution has occurred in mass spectrometry as a consequence of electrospray and MALDI and HPLC. And then, you know, I was an old sector instrument person. I loved sector mass spectrometers. And so when I came here, I had a 4-sector instrument of all things. And of course, all of that technology has disappeared, rightfully so. And it's become, you know, ion traps and or B traps and QTOFs and some Fourier transform ICR instruments. So it's been really an exciting life for me because, you know, I went from taking mass spectra on a chart recorder to watching my students take, and that spectrum would take 20 minutes or 30 minutes, during which time you could have a coffee or a cigarette or whatever your proclivity was, and think about your data. And now, of course, in 20 minutes, you get 20,000 spectra a second. So it's just an amazing revolution. So I was there for the whole story when to get a high resolution spectrum of a mass spectrometer, you took the data in the computer and then you output it on a paper tape, and then you read it back into the computer to process it. So, and that was actually in the first 5 years of my career. So it's been quite a run. I was hoping to have, yeah, here we go. So can you see my pointer?
Yes, we can.
So this sort of looks like Washington University, not this time of year, of course. But it's our signature building, and then I stole your icon. So this is a story. Tonight's lecture is a story about protein higher structure determination. So I want to put in, give a shout out to Roger Liu and Don Rempel, who have co-authored a paper which is coming out in Nature Protocols on what we call FPOP, fast photochemical oxidation of proteins. Sure. So we like the abbreviation FPOP. In fact, my students violate this kind of word. They have turned it into a verb. I'm going to FPOP a protein, they say, which is perhaps not the intention of the word. It was supposed to be a noun. Fast photochemical oxidation of proteins. So you might look for this. It's coming out very soon. And this just came out. So again, Roger Liu. And Mong Ru, who just finished her PhD and is now at Seattle Genetics, notice here that this review talks about all kinds of chemical modifications of proteins, including cross-linking and including studies dating from the 1960s when I was a postdoc. And notice that the review is about 100 pages long. So it's a very thorough presentation. It just came out in Chem Reviews. So it's a very comprehensive review. It includes HD exchange, includes chemical footprinting, includes some cross-linking, and we tried to tie the whole thing together as a way of doing, for example, HDX to be used as a footprinter, or free radical footprinting with different kinds of readouts. So there's Roger and Mong Ru, and we were together at, it seems like an eternity ago, but it's just 11 months ago we were at Lake Louise for the Lake Louise conference, which I highly recommend if you've never gone. But you have to drive from Calgary up the hill, up the mountain to Lake Louise. It's a beautiful place. Anyway, there was a nice conference there that we attended. And so that's Roger and Mong Ru who helped me with this Chem Reviews review. So what motivates our work is this triangle here, or maybe you can think of it as a pyramid if you want to think three-dimensionally. And at the top of the pyramid is NMR and X-ray, because these are the gold standard structural tools that can be used in protein structure. They give coordinates of all of the atoms. Cryo-EM has rightfully taken a place among those three methods. And at the bottom of the triangle are traditional tools like circular diachroism, optical methods, IR, UV, light scattering, and of course, separation methods like chromatography, which also tell you something about higher order structure, but they don't give often any kind of regional or even amino acid specificity. So my goal is to place mass spectrometry, to wedge mass spectrometry in this triangle below NMR and X-ray, because we're not ever, well, I shouldn't say we're not ever going to give coordinates. We could maybe by modeling, but we can give structural information, principally in a differential way. So the bottom of the slide sort of motivates, little biochemistry one presentation. It motivates what people do. People want to know about primary structure. And of course, the way you learn about primary structure today is by doing proteomics, right? And you can learn about post-translational modifications, something that Ben Garcia does with a flare. And our emphasis, however, is secondary, tertiary, and quaternary structure. So we want to take the information that you can get from primary structure proteomics. We want to make sure that that information is complete with high coverage, and we want to make differential measurements. We want to differentiate, for example, a protein in an unbound state with a protein in a bound state, an apostructure from a holostructure, or a mutant from wild type, or a protein at pH 6 versus a protein at pH 7. So you get the idea. The idea is to make differential measurements, to compare a protein from in one state versus a protein in another state. So this, of course, does not give you coordinates, but it gives you a comparison and allows you to learn about higher order structure, which I think many of you know is really a key thing as the world of pharmaceutical and biotechnology turn to proteins as pharmaceuticals, because you want to know, of course, that whether the higher order structure has been preserved. So that's just one application for higher order structure. You want to make sure that the compound, once stressed, for example, if it's changed its higher order structure, can you report on that somehow? And of course, you can use these tools to report on higher order structure. But I think you can use, and that's what the theme of the lecture will be, you can use mass spectrometry-based methods to zoom in and get some spatial resolution. So it's a very simple idea. So we can use hydrogen-deuterium exchange for that. Some people would argue that I shouldn't call that footprinting because hydrogen-deuterium exchange also tells you something about dynamics, and I agree, of course. But, you know, one strategy with HD exchanges, for example, and I'm going to talk about this, is epitope mapping with antibodies. And so you can do the HD exchange pattern of a protein with and without the antibody, and of course, learn what the binding site is coming up. So protein footprinting, that's one way of doing protein footprinting in a very rapid way is this FPOP, where the experiment is designed to use a flow system. And in the flow system, just a standard syringe pump. You intersect that flow with a laser. In our case, it's a krypton fluoride pulse laser. Some people in industry say, well, that rules out for me. I can never put a laser in my laboratory. Our safety officer won't allow it. So there is a company now that's trying to do this, and it's successful so far in doing this with just a discharge lamp. So you don't need the laser, although I think for an academic research lab that's trying to do the problems that I'm going to tell you about, this is really an important thing. And then the footprinting takes place here at the junction of the laser beam and the flow beam, the flow tube, and that sample then flows on. It's been footprinted, it flows on and is collected. And then cross-linking, I would argue, is another way of footprinting. It's a way of doing two footprinting reactions, one on one protein, the green one, for example, and another one on the purple protein here. And of course, the cross-links tell you that, for example, this residue is close to this residue, which is really important. So there's a footprint here and a footprint here. And of course, the way they connect is what gives you the information. I call that all protein footprinting, and that's why it's many of these ideas are covered in that review, which I would recommend to have a look at it. So a little more detail about FPOP, since that's going to be a big theme of this lecture. So here's the flow tube, and here's a protein in the presence of H2O2. So we have to introduce some radical precursor. So indeed, H2O2 could affect a protein, could oxidize it, but that oxidation is slow compared to what we're going to do. So that solution flows past a laser beam. The laser beam is perpendicular. This is this junction here. So I've just amplified that junction. The laser beam intersects with the flowing protein. It converts the blue dots, which are hydrogen peroxide, into red dots, which are reactive OH radicals. So we time the firing of the laser beam and the flow to permit an exclusion volume. So this, let's call it a plug, an irradiated volume. This volume was a moment ago here, and now it's advanced to this point. Another insight that I can provide is, of course, you can think of this as a reaction chamber. Huh? In this reaction chamber that's been illuminated by the laser, you get chemistry happening, and the chemistry footprints the protein. And between this, which is now about to be irradiated, and this, which has been irradiated, and this, which was irradiated a moment ago, is an exclusion volume, which allows you to have a little space between them so that the idea is to prevent any double hits. So you don't want any significant double hits. So I think that's really a key element of this design. It prevents double hits. A second key idea of the design is this flowing solution has a scavenger in it, usually a single amino acid. And that single amino acid scavenges the OH radicals and limits their lifetime in this plug. So it's designed in such a way that the lifetime of the radicals is 1 microsecond, thereabouts. Yet the yield of the radicals, I think, is really quite remarkable. It's 1 millimolar. So you make in that solution a transient concentration of 1 millimolar radicals. So by the time this plug has moved even a fraction of a millimeter, the chemistry is done. You know, it's over, 1 micromolar. of 1 millimolar of radicals has disappeared to some very small 10 to the minus 8 molar concentration. And effectively, there's no more chemistry. So the protein's about a micromolar, sorry, 1 to 10 micromolar, even less. And the H2O2 is about 10 millimolar. And once the laser fires, we've done dosimetry, the yield of the radicals is 1 millimolar. So you see there's plenty of radicals. Micromolar protein, so there's 100 times, 1,000 times more free radicals than there is protein. So it's never going to label the whole protein, but it's going to label in general. It's going to label a large number of amino acids with this reactive species here, OH. So that's David Hambly, the person who invented this in my lab. And he graduated in 2006, so this is 14 years ago. He went on to Amgen, and he's now moved on. He's a private consultant working on biotechnology and protein therapeutics. So David was a young father here, so here's how he did it. I love this slide. You see the 150 micron tube placed in Legos. Here's the exit of a krypton fluoride laser. So the laser beam goes across this way. This is the beam stop here. You can see it's been beat up. And the chemistry takes place right here where my laser pointer is. And then this tube, the syringe pump is back here. And there's a collector over here. And so this flow goes into the collector. And in the collector, we add some catalase at the end to get rid of the H2O2. So the nice thing about this, by the way, is that unlike HDX, one can put the sample in a freezer, and work it up later on. You don't have to do it now because the imprint, the footprint, is permanent. It's been imparted on the protein, and there's no back exchange like there is with HDX. So you can be leisurely about it. You can accumulate all of your samples. You can do some detailed study. And hold on just a second. We have groceries coming. I'll be right back. So my wife went for a walk because she didn't want to listen to me yelling at you on my Zoom session. And the groceries just arrived, so there's no one to pick them up except me. Sorry, just had to let them in. So what more to say about this? So of course it looks a little more elegant now. It's not made up of Legos, but you get the idea. So it's now there's a set of optics there that make this allow us to focus this more easily and more readily. So this just emphasizes the point that here's the optics, here's the syringe pump, here's the exit of the laser, and in here, not very visible, you can see it here, the capillary tube, like you would use in nano LC, 150 micron tube. And then we just use a sign here sign to tell us where the laser beam is. So it'll make a little burn mark. So this is not really an emphasis for my students to sign something. It's just they can look at the beam and they can see the capillary. And you can see that the beam is irradiated and the irradiation zone is about 2 millimeters. So that's the whole story. So what does it do? Well, without belaboring the point, it The OH radical takes off a hydrogen atom. And here I show it for proline, very inert amino acid. And that free radical then is quenched by oxygen, which is dissolved in solution. It's always there. So you can do this in a cell, as my former postdoc, Lisa Jones, who's at the University of Maryland, just nearby you guys, is doing. She's footprinting using FPOP in cells and in small animals, like C. elegans, in the same kind of a flow system design. So she's taken this idea to the next step where one can do protein footprinting in cells in vivo. The ultimate goal, the ultimate outcome is to put an OH group where you formerly had an H. And of course, as mass spectrometrists, we all know that that's very nice mass change. Hydrogen has become an OH group, so that's a plus 16. Well, really it's 15.9949. So the beauty of all today's instruments is that we can use 15.9949, so we're not misled. We can search for it. And the difference between our analysis and a proteomics analysis is that we want high coverage. So we really have to work hard. Because of course, if we say our footprint tells you that the binding site is here, and if we only covered half the protein, of course, that could be misleading. Sometimes you get other modifications like plus 14. And these are all detailed in a review article that Mark Chance wrote in 2007. So Mark Chance was the person who first called this protein footprinting, and he used OH radicals that are formed on a synchrotron. So I don't want to demean that, because I think it's really an important advance. The nice thing about FPOP is you can do it in your own laboratory, of course, because you don't need a $1 billion synchrotron, and you don't have to queue up and go to Brookhaven or Argonne to do it. So there actually is a beamline in Brookhaven that's reserved for protein footprinting that you can use if you contact Mark Chance. So the rest of the lecture is going to be 5 or 6 or 7 little vignettes about how we can use this idea. So I'm really not going to try to tutor you. My goal is to expose you to some ideas that maybe will be relevant to your own research, where you can use FPOP, HDX, and other kinds of footprinting for solving some biochemical problem. So one biochemical problem that's really important in biotechnology today is antigen-antibody interactions. And I think you all know what that is. So here's the antibody, and the region where the antibody binds the antigen, here's the antigen, is called the epitope. And of course, that's a really important, so you'd like to know, for example, how the antibodies that Donald Trump took, how would they bind, for example? You can be assured that somebody has done something like HDX, or they've done biochemical assays like mutagenesis to figure out where the binding site is. So this is not COVID, this is malaria. So malaria is not, most of you know, I'm sure, malaria is not a virus, it's a parasite. And it has on its surface Duffy binding protein here in green. And it binds then here to an antibody. There's an X-ray structure here. My collaborator, Raj Tolia, who's now at NIH, used to be at WashU, has provided these materials and is an X-ray crystallographer, works on virus proteins and parasite proteins. So the contact residues tend to be loops. They tend to be on the surface. I guess that's no surprise, because that's where the antibody is going to interact with the protein. And so HDX is really, I think, very conclusive. Here is a story of 3 antibodies with the Duffy binding protein in malaria. Here are the names of the 3 antibodies. Don't ask me to defend these names. I don't know where they come from. 2D10, 2H2, certainly nothing like naming alkanes. And here's some of the peptides in Duffy binding protein. The coverage we usually get for HDX, and here's the name Tolia, by the way. The coverage we get with HDX is often very close to 100%. So that's cool. You have to have that. So here's a peptide from that protein, 408 to 413. The blue is the unbound state. The red is the bound state. And you can see there's a really nice systematic difference. There are error bars here, and they're at the size of those points. So the precision is really good. And you can see that 2D10 and 2H2 bind at that site. And here's a larger peptide. It's nice to get redundant information. It's +5 because it's a bigger peptide, but it's also recapitulating the differences that are seen here and maybe telling you that there's some additional binding in that region. Now 2C6, which is hard to see, but I hope you can see it. You can see that there's no difference. And so 2C6 binds differently. In fact, 2C6 binds at 471 to 478 and 479 to 484. So you can see very clearly its differences. If you've never seen these before, I think they're really beautiful to see because it shows you just where the binding is. And so this is a region that doesn't bind to either one of the antibodies. So 90% of the peptides that you see in this experiment look like this. There's no difference between bound and unbound. So it's very convincing that this is giving you the epitope is maybe not amino acid by amino acid. The way an immunologist would do this, they would change specific residues into alanine, which allegedly is not binding. They would change them into alanine, and then they would call that an alanine map, or sometimes an alanine shave map, where they would change more than one of them. And of course, that would be very convincing if you changed a critical binding residue that was, say, tryptophan, and you changed it to alanine, and the binding went away, then you could be really convinced that you had specific amino acid binding. And of course, the way Raj Tolia did it to help us confirm that these HDX results are okay was by X-ray crystallography. So there are the epitopes, and you can see that very clearly. So here is a collaboration with Bristol Myers, not so far from the Delaware Valley. And Bristol Myers did it with alanine mapping. And so I'm just showing you the regions. I'm not going to show you the data. Here's HDX. So HDX overlaps. Of course, it has peptide level information, so it's not so sharply defined as here. But you can see that there are peptides that overlap what the alanine scanning tells you, and that there's also a region that, and this region, by the way, is not showing binding, but is showing deprotection. So it's a remote conformational change, of course, which is not being picked up here. So you get not only the binding, but you also can learn something about maybe allostery or remote conformational changes. And now with FPOP, you can see that there are two peptides and they basically overlap with this one and with this. And here it is again, overlap, overlap. And then this is slightly different, this region where you have a decrease in protection, a conformational change that makes the protein more solvent accessible at this region. So I think aggregating proteins are really a challenge for the biochemistry community. And so we've tried to dig in with aggregating proteins, especially amyloids. Amyloid or aggregating proteins are also important, of course, in biotechnology, because you don't want your therapeutic protein to aggregate. With amyloid proteins like A beta, like synuclein, like tau, these are proteins that are commonly studied by what's called THT analysis, where you put in an organic dye, and the organic dye gets immobilized in the aggregating protein once it starts to form protofibrils and amyloids. And then because of its immobilization, it will fluoresce. And so you'll see the fluorescence increase. with this really characteristic sigmoid curve, having a saturation phase, an intermediate growth phase, and a lag phase. So, you know, that's a structural tool, obviously, but it's not telling you much about information at the residue or amino acid level. So here is A beta with a THT. So you know that it's aggregating. That's 1 to 42. The more toxic of the various A beta that are formed in the human brain. My colleague, Carl Frieden in the medical school, came up with a different way where he attaches a molecule to A beta, and I think that's a little bit dicey. So I'm not talking behind his back. I've said this to him. It's a little bit dicey because, of course, A beta is very, very sensitive to modifications. So its aggregation behavior is, if you put in a marker, it could affect the aggregation itself, because just think about A beta 1 to 42, it aggregates much more rapidly than A beta 1 to 40. So just 2 amino acids make a big difference. So HDX recapitulates this. So this is HDX, but it's at the peptide level rather than at the whole protein level. So we published this a couple of years ago. I'm not going to dwell on it, but you can see that this TMR curve, which shows what happens to the monomer here is becoming dimer, and then it's becoming, goes through a lag phase, and then it goes through a second sigmoid. So the TMR pulls that out. HDX pulls it out too. So here's monomer going to dimers, and then a lag phase, and then the sigmoid, and then the saturation phase. But it pulls it out at the peptide level. So you're getting regional specificity with HDX, and you're not modifying the protein. So there's no modification of the protein except to put it in D2O, or to put it in FPOP. So here it is in FPOP. Work of Kelly, who is now at Genentech. So this is the control experiment, just the A beta by itself. No FPOP, it's just in H2O2. And there's the unmodified, you know, the protein is 4,000 something molecular weight. So this is the +5. So it's a clean, nice clean spectrum. There's some modifications, maybe some little basal oxidation going on here, but generally it's quite clean. And now when you submit it to FPOP immediately, right after you put it in solution, after you've monomerized it, put it into solution, you can see that it's really intrinsically disordered. I mean, the FPOP pattern is so clean and clear on that question. You can see that it's +16, +32, +48, +64. It undergoes lots and lots of modification in this very complicated pattern because it's intrinsically disordered. It doesn't have much of a high order structure, and so it labels everywhere. Now, as it aggregates, the pattern becomes simpler. So this is 40 minutes, this is 15 hours, this is 24 hours, this is 48 hours. You can see how it simplifies. So, I mean, this to me is really convincing how FPOP is so sensitive to the state of the protein. Here is an intrinsically disordered protein that has found order by aggregating and has protected itself. And it's now much less surface accessible. So, okay, now we can dig in and look at peptide level. First of all, we plot the data here. So there's Kelly's name, published this 4 years ago. So you see, it's even more informative. You get a sigmoid, a second sigmoid, and a third sigmoid. So I don't know how seriously we want to take, but this is really small, monomers, dimers, and then they begin to aggregate, and then they begin to make protofibrils. And these protofibrils go into larger protofibrils and suddenly become organized, and then they make a fibril, an aggregate. So FPOP gives you more information than the THT or the HDX. It gives you a more informative sigmoidal behavior that of course you can now check what you can use this as a standard. You can compare how does this curve behave if you put in an inhibitor, if you put in an antibody that's supposed to clear it away or stop it from aggregating, and so on. So no, moreover, you can dig in at the peptide level and even at the residue level. So this is the peptide level. So we've digested A beta, the small polypeptide, and the digestion gives us 3 pieces. It's not trypsin, it's asp-N, I think. I'm sorry, I'm blanking on it. Anyway, look at the N-terminus. I mean, it seems to have the elements of those sigmoids, but there's almost no change. So the N-terminus of the protein, as it aggregates, is just remaining solvent accessible. So it's like, you know, it's sticking out into solution, and no matter what the aggregation state is, it's solvent accessible. Whereas the second part of the protein, the blue, undergoes this classic sigmoidal behavior. Here we just fit it to the same sigmoid we fit before. So you can see that the fit is pretty good. And the C-terminus also, notice that it goes from, say, well, this is, I would have to show you the other one. So this is, I need a differential experiment coming up. So here's the C-terminus, and so the C-terminus also fits nicely to 3 sigmoids. And it goes from, it's also involved in binding because it's going from about 60% initially. No, that's about the same, 60% initially, and then going through this sigmoid behavior. So now you can dig in further, and you can look at individual residues. In principle, what we would like to do is develop methodology that you could look at all residues. So FPOP in principle is reactive with something like 14 amino acids. But you can't really do 14 amino acids because some are more competitive, like histidine and phenylalanine, react much more readily with OH radicals than do, for example, glycine and alanine and valine. So the information will be selective, even though it's a generally reactive free radical. But you can see that this histidine is not being affected at all by the aggregation. So it's sticking out in solvent throughout the whole aggregation. And it's polar, and that's probably reasonable. And here's histine 13, and it's the same. Whereas I think the culprit in the aggregation is really phenylalanine, these hydrophobic residues. And the reason we're equivocal here on F19 and F20 is that we're not sure which one it is. So this is an MS-MS experiment coupled with extracted ion chromatograms to see this particular specifically modified amino acid in a peptide. And you can see that that's giving us this same behavior as the whole protein. So this particular region recapitulates the whole protein. So I think this is a really important region in the aggregation. And it makes sense because it's hydrophobic. And here M35 also, methionine is very reactive with radicals. You can see that it also is also involved in the aggregation. So there is a solid state structure by NMR and by cryo-EM, and that's the way it looks. You know, you see the fibril looks like sort of a noodle, and sticking out from the noodle are these N-terminal amino acids, N-terminal peptide and amino acids that are solvent accessible and undergo modifications. So as I mentioned, we can use this for looking at inhibitors, and we've just started doing this. So here is the effect of adding an inhibitor. The inhibitor is curcumin. It's an interesting choice. It really doesn't take a lot of brilliance to pick curcumin. Epidemiologists know that Indian people do not suffer the high incidence of Alzheimer's disease as do Europeans and Americans. And one thought is it's in their diet, that all of their life they eat curry, and curry has turmeric, and turmeric has curcumin in it. And curcumin is a polyphenol, and you can see that curcumin is binding to A beta. Here is A beta itself, You see, it's highly modified and now it's highly protected. So you can learn where the binding site is. Now, I don't know that it's a good inhibitor because really the sigmoid curve is the reporter on aggregation, and it hasn't changed really too much, a little bit. But certainly you know the binding site. So I'd like to say that this kind of a procedure would be very important in aggregating proteins because it can tell you what are the cores of the aggregation, what are the key residues, the key sites that undergo the aggregation. And then if you use an inhibitor, does that inhibitor affect the aggregation? Where does the inhibitor bind? And so these are difficult experiments because, you know, this system, after say 900 minutes of aggregation, is really heterogeneous. It has, you know, 100 MERS, 101, 102, 110, 120. There's all kinds of aggregates. So you can look at the starting state by NMR, and you can look at the final state by cryo-EM, but FPOP gives you, I think, a good view of what's happening in between. So we're coupling this with DFT. I'm not going to go into this very much. DFT verifies that the curcumin, this is curcumin, and this is A beta. So you can see it has some higher order structure now. And I think that's been induced by the binding, and the binding is at the E terminus, just as FPOP said. So determining binding affinity for signaling proteins is another application. So this is the third idea that we can explore. So this is, of course, very important in therapeutics. You want to know where a small molecule binds to a large protein. So we can do this by HDX. 16 years ago, we published a paper we called the method Plimstex, where we titrated the protein, that's this axis, and followed HDX as the indicator, you know, perhaps the most expensive titration ever, using mass spectrometry as the indicator. And we do that at a constant time, so we're not following kinetics, we're following a titration. And you can see the protein becomes protected as you titrate it with a potential ligand. And you know what region it's becoming protected. And you can fit this curve to say that the binding affinity is 6 micromolar. So this was measured by fluorescence by my collaborator at WashU Medical School. And he got 5 to 10 micromolar and we got 6. But unlike fluorescence, we can tell you, here's the region where it's binding. Whereas other regions that are not binding give you flat curves. So the rest of the protein looks like this. We get, of course, as I mentioned before, we need high coverage. I'm not showing you the high coverage, but you can imagine the rest of the protein looks like this. There's no change. These are the 3 regions which must contain critical binding residues that, and this is, by the way, a protein related to A beta. This is ApoE, binding with a molecule that is intended to modify its behavior. So this is FPOP doing the same thing. So the protein here is calmodulin. It's been loaded with calcium. So it's calcium-bound calmodulin. And calmodulin is a signaling protein. When it gets calcium, which is its message, it then will bind to melatonin. And the literature says that the binding constant is on the order of low nanomolar. So now we're titrating it in the same way. We're titrating it with calcium, with melatonin. We bind it with calcium and we titrate it with melatonin, and we're following the titration at the peptide level. So here are just 4 peptides, and you can see clearly this is a classic binding behavior. So 14 to 30 is involved in the binding. And by the way, that's in a hydrophobic region of the protein that becomes exposed with calcium binding. And so it's now set up to bind melatonin, which is a very hydrophobic model peptide. Whereas 38 to 74 and 91 to 106 are not binding, whereas 127 to 128, 148 is also a binding region. And we can fit these curves also, get this. So this is the fit. it says that the binding constant is 2 nanomolar, so it can be very sensitive. Now we step back one step back, and we look at the calcium binding. So this is the signaling, right? This is the protein binding to calcium. Calcium is the message. And so when calcium binds, and actually 4 calciums bind calmodulin, we can track the protein region by region. So first notice that we're looking at the N-terminus, and then we're working our way across the whole protein, region by region by region, titrating the protein with calcium and using FPOP as the indicator. So 1 to 13 and 31 to 37 is essentially not much is happening to the protein. And so a signaling protein, as you might guess, is undergoing lots of contortions, lots of different interactions, lots of different conformational changes as it binds. So let's look at a binding region. So this pink one is a classic binding region where the protein, as it picks up calcium, it becomes protected. And the amount of FPOP decreases in this nice sigmoidal kind of curve, where we could use this curve to calculate the affinity. So we know that that's a binding site. So here's another site where just the opposite happens. 38 to 74, it begins to become exposed as you titrate with calcium. So it's a segment of the protein that's undergoing 2 different kinds of conformational changes. It's opening up, as you see, becoming less and less protected. It's reacting more and more with the free radicals. Then it goes through binding. It comes down, and then it opens again. Really, I think, very fascinating what's happening to this protein. So here's a region, I would call this a region in the middle of the protein that's allosteric. So as calcium binds, this region opens up, and it's part of the signal. This is where melatonin was binding in the previous slide. So the melatonin binds here because this region, which is buried in the apoprotein, becomes exposed, and you can see it very clearly becoming exposed. So here's a region which shows binding and then exposure. So we can learn about allostery, we can learn about cooperativity. Here's a region, and you can compare these regions one with another. You can see what's happening to this region as binding is happening to this region. So I think it's very powerful to allow you to sample what's happening to the protein as a function of its binding. So when we look at the onsets for these binding regions, the protein has 4 binding sites for calcium. They're called EF hand 1, 2, 3, and 4. And you can see the first binding, the first EF hand 4, the first one to bind is this one because its behavior, it changes its behavior. It goes to a binding state first, followed by the pink second, followed by the green third, followed by the red fourth. So there's the order of binding. EF4 binds first, 3, 2, and 1. So just to summarize, this gives you binding sites. Processing these curves with our model gives us affinity. You can see the order of binding here. You can see allostery, as I showed you in the previous slide. And you can see how one region in its binding cooperates with another region. So I want to show you this experiment. You see how we're doing for time. We're running out of time quickly. So this, I think, is really a nice recommendation for FPOP. So let me show you this. We want to use FPOP for protein folding. And what motivates that decision is that FPOP is really fast. I told you before, it's microsecond labeling. So you can take a snapshot of a protein, say in its unfolded state, or in an intermediate state, or a folded state, and you can do that on the microsecond timescale. So protein folding is the purview of a lot of very sophisticated experiments in stop flow and modeling and so on. And I think this kind of an experiment with an FPOP FPOP apparatus would allow a lot of people to do protein folding and use it to answer questions about proteins that don't require a great deal of sophisticated apparatus. Well, you may argue this is already a 2-laser experiment, but it's not that complicated, really. You have an excimer laser that's already here for FPOP, and you have a YAG laser that in our first experiment, we frequency shifted the fundamental of a YAG laser to 1900. And the reason we did that is 1900 is resonant with water. It's resonant with a bending motion of water. And so the light energy will be absorbed at 1900. So this is the pump laser. This comes in and pumps the water, raises the temperature just at this junction by 10 or 20 degrees. It's amazing. Huh? And then the excimer laser, timed to follow, shoots and makes OH radicals, which in a microsecond footprint the protein. So watch the 2 lasers. Oops, wrong way. One laser fires in the other. And so we just changed the time with the signal generator and the delay circuit. We changed the time between the 2 lasers. I'll skip that and show you what happens to this protein, which, by the way, at low temperature is unfolded. It's a strange protein. It's called bar star. Turns out that almost all proteins will cold unfold. Most of them cold unfold at temperatures that we don't care about. But this one cold unfolds at about 0 degrees or 10 degrees. Here it is unfolded. Look at that. It's just labeled everywhere because it's unfolded. So it just underscores the sensitivity of FPOP to a disordered protein. It's made disordered because it's unfolded. And here it is folded. The control, the room temperature control, the protein is folded. And so you can see the FPOP pattern is quite simple. The peaks are hairy because we're seeing all the isotopes. So don't worry about that. So there's plus 16, plus 32, plus 48. But contrast this very simple pattern with this very complicated pattern for FPOP. And then notice that we can explore time of folding by changing the time between firing one laser and the time for firing the other. So here's 200 microseconds, 400 microseconds. You can see that time is sufficient for the pattern to begin to simplify, to simplify, to simplify. So if we just take the centroid of all those data and plot it, the centroid is becoming less, and so you can see the protein is, we're using the mass spectrometer just to weigh the protein, and the unfolded protein, of course, weighs more than the folded protein, and so you can follow the centroid and get the rate constant. It's 1.5 per millisecond, comparing with fluorescence data in the literature, which is 3. So I think it's good. But now we can dig in and look at the residue level. So look here, here's 2 residues, very polar, E57 and R75. Here they are in the X-ray structure of bar star mapped. You can see them sticking out in the folded protein, and in the unfolded protein, they would be in a similar situation. They're sticking out into water, they're solvent exposed, and their reactivity doesn't change with folding time. Remember, that's the time between the 2 lasers. So as you increase the time between them, the amount of labeling is the same. So the red point is the final point, and all of these are time-dependent data. Well, something better changed, right? And so here's the change. Histidine, leucine, and leucine, very hydrophobic amino acid residues. They're changing. They're becoming more protected as the protein folds, more protected as the protein folds. So you could get a rate constant for this. The other point to notice is that these are really not very reactive amino acids. Huh? Leucine, I mean, how are you going to footprint leucine, for goodness sakes? But a free radical will do that. So the free radical is reacting with leucine, a very hydrophobic side chain, and it's footprinting it, and it's telling you that 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 residue is involved in the fold. And when you look at the X-ray structure, you can see where they are. They're in the center of the protein, and they form the hydrophobic nucleus around which the protein folds. So these residues buried in the hydrophobic core of the folded state, and they're involved in the first stage of protein folding of this particular protein. Residue level data, and hydrophobic residues, residues that would be very hard to modify chemically unless you chose a free radical. I'm going to skip this because we're running out of time. Our new emphasis, just give me a few moments if you could. Our new emphasis is membrane proteins. We think that protein footprinting of membrane proteins is going to be our new holy grail. So far, we've learned that if we put our protein in a lipid nanodisk, these are nanodiscs promoted by Steve Slygar at the University of Illinois. Here we put in that nanodisk a reaction center in photosynthesis. So we had, for many years, a very strong collaboration with Bob Blankenship here at WashU. Bob just recently retired. OH radicals don't footprint the transmembrane region. They footprint only the external part. So we just published a paper recently. It's come out in PNAS on KRAS protein, a protein with big implications in cancer. It's not a membrane protein, it's a membrane-associated protein. And the hypothesis is it becomes a membrane-associated protein by a very hydrophobic C-terminus that sticks into the membrane and makes the association. So it's not transmembrane, it's membrane-associated. I'm going to skip that. That's just the, and just show you the residue and peptide level bases. So here we're getting kinetic data just like you do in HDX by varying the scavenger concentration. So this is not time. This is the modification of a reporter peptide that's counting the number of radicals that we've made. And so you can see the difference between the protein in the absence of a nanodisk and the presence of the nanodisk. Red versus blue is the same, is the same, is the same. These are regions of that protein that are not associated with the membrane. But if you go to the C-terminus, you can see that the peptide in the presence of the membrane, presence of the nanodisk, where it's now associated, is protected. So this is the region that's now associating with the cell membrane. Of course, we're not doing a cell membrane, we're doing a lipid nanodisk. But in principle, you know, Lisa Jones could do this, you know, in vivo. So this is still in vitro, but it's one step closer to in vivo because we're using a nanodisk. So you can see that the binding is at the C-terminus. And then if you look at the residue level, you can see that that information is recapitulated here. You can see that these residues change because they are protected by the binding in the lipid nanodisk. I'm going to skip this in the interest of time and just mention to you that the story is not over for free radicals. So this is the work of Ming Cheng, who's now at Scripps. He just finished his PhD a few months ago. And he made a connection in his research between trifluoromethyl radical chemistry, which is used in materials science, with our area, which is structural proteomics. And he built the bridge by using this radical, CF3. So the story is not over. OH radicals are not the only radicals that one can use for footprinting. So CF3 is a very interesting radical. It does not react with methionine, which is the most reactive residue with OH, or with cysteine, which is the most reactive radicals with OH. It doesn't react with those, yet it reacts with something like 14 amino acids. So it has complementary chemistry. I'm just going to show you how you form it. Here's how you form the CF3 radical. So what Ming did is you can buy this reagent. This is trifluoromethyl sulfinate. So it's a salt. And it'll react with OH. The OH radical will come in and attack the sulfur. and push off CF3. And I think this CF3, it wakes up in water and it says, oh my God, what am I doing in water? I'm highly hydrophobic. This is not where I want to be, but its precursor, you see, is very water soluble. So it's okay here. Now it's in a hydrophilic medium and it's very hydrophobic and it reacts really like crazy with proteins. So there's all kinds of opportunities, I think, for reactive species. Not only radicals, but carbenes, but carbocations maybe. We're starting to work on that idea. So there will be other kinds of radicals that one can design, that one can choose to answer specific questions like I've tried to answer in tonight's lecture. So it's a very reactive, this just reinforces what I said. It's very reactive. It doesn't react with methionine and cysteine, which is good news, I think. And it reacts with some things more readily than does the OH radical. So it has complementary reactivity. I'll skip that. This is the summary. So you can use these ideas, both HD exchange, and FPOP, for mapping epitopes, for following amyloids, for determining binding affinity at specific regions of a protein, which is very nice, I think. I think what holds this back is that there's not a lot of software available. Our software is not designed to be friendly. And so I'm trying to talk protein metrics. We're working with them to try to put this kind of software in their packages. We can follow protein folding in that 2-laser experiment, which I think could bring protein folding more into the mainstream. If you have a new protein, you want to know something about it, its fold is important to know. And so I think a mass spectrometry experiment can give you that. I skipped this, but protein conformations that are hidden in X-ray crystallography and in NMR can be revealed by FPOP. And I think our new holy grail is membrane proteins. So we're just beginning to work very hard on membrane proteins. I showed you that we could do membrane associated proteins in that recent paper. And our goal is, to do transmembrane regions, the footprint transmembrane regions of proteins. And this can all be accelerated and be made more informative by having new reagents, which is, of course, chemistry in action. So here are the people that did the work. I won't go over them one by one because I've taken up too much of your time already. But this is my group Christmas a year and a half ago, almost 2 years ago. It's shrunk a little bit, but I thank them, of course, for all of their hard work. I'm just here to do the public relations. And of course, I thank Gen X, and that's the company that has sponsored some of this work. They're doing they're developing an FPOP apparatus that does not need a laser. And of course, I thank you for listening. Thank you very much.
Well, thank you very much, Mike. That was a wonderful presentation. I'll, at this moment, open the floor up for questions. If anybody does have questions, please feel free to go ahead and unmute yourselves, or you could send them in the chat as well.
See anything in the chat? Yeah, Tim.
Does anybody have any questions or comments?
There was too esoteric or perfectly clear.
I think I would lean towards perfectly clear there, Mike. I think it was a wonderful talk. What would be, maybe this is a bit forward looking, and I don't know if you could even comment on it, but I guess what would you say is the outlook, or what are you guys working on, or maybe you can't say, but in regards to future reagents to use? I know that it looks like you're using primarily 2 reagents now. Do you see that field significantly growing in terms of, a variety of new reagents coming about in the near future?
I skipped over this, but I'll mention it because this is en route. And a new reagent. It's an old reagent, the OH radical for membrane proteins, using material science to attach a nanoparticle to a membrane. And then with that same laser, we can make, at the vicinity of the membrane, OH radicals. At the same time, we do a photochemically allowed reaction of a 2+2 cycle addition where we add acetone to a double bond. You know, you're going to find double bonds in membranes. And we're going to make a four-membered ring. This is called oxidation. An oxidation ring. And that's going to kink this membrane. Like this, huh? And I don't know, this is our cartoon. This is probably too big a kink. So at the same time, imagine you make an OH radical and you perturb the membrane at the same time. And the OH radicals, we hope, will enter the membrane and footprint the protein. It's working. So in the absence of the cycloaddition chemistry, the absence of kinking the membrane, the only reaction of OH radicals is here on the extramembrane part. Well, this is explosive solvent, right? No surprise. But our little chemistry trigger will open up the membrane, lets the OH radicals inside, and now look what happens. These are all modification sites in the membrane. So I think there's two things we can do. We can use ideas like this, open up membranes, they take advantage of the radicals we already know and love, or we can generate new radicals. So some of the new radicals that we're using for membrane proteins are fluorinated alkyl iodides. So we're choosing them because they're hydrophobic, so we think we can put the reagent in the membrane. It's an alternative to this idea. We want to put the species in the membrane. It's hydrophobic, not hydrophilic. We urge it into the membrane. Gentle sonication. And then we turn on the laser. And the laser homolyzes the carbon-iodine bond. It makes an iodide radical, which footprints the radical. So that's another idea. So the idea is to put into the membrane reagent that's hydrophobic, where it will be very happy in the membrane. And then you use this photochemistry to make another radical, iodide, which now has no choice but to interrogate the transmembrane region. What do you do here? Please don't take this up. This could be me. So that's. I think those are the kinds of ideas we want to pursue. We want to pursue new radicals. I think there has to be more radicals than just CF3. So we use the same platform to make carbines, and we've done that. The carbines can also be made on this platform. So you can choose a carbine, which is very reactive. We think carbines can react in nanosecond time frames. So you could use the same idea. You could insert a carbine precursor into the membrane by just tailoring it so it's hydrophobic. You need to have some organic chemistry skills. Drop it into the membrane, and then in this flow system, taking advantage of the same flow system, flow the system past the laser beam, which activates the radical precursors in the membrane. So I don't know if you know, but something like 60% of the drugs, small molecule drugs that are used today, target membrane proteins. Yet we know there's only 100 crystal structures of human membrane proteins. So it's an area that's just waiting to be explored in terms of dynamics, in terms of printing. A little bit of money from the American Heart Association to look at some membrane proteins that are important in heart disease. So that gives you a little idea on what radicals we can use. Iodide radicals, carbonate radical anions that are not so good. Carbonate is -2. If you oxidize it, you get carbonate -1, and then an unpaired electron. Those radicals are biologically significant, probably, and they're not particularly interesting as foot printers. But anyway, there's a whole world, I think, of radicals and carbene, maybe even carbocation, maybe even positive species. And you could generate with a burst of a laser and then do really fast foot printing.
Great. Sorry to cut you off, Mike. We're having some reverb on our end. Perhaps maybe it's that your audio is up too loud and it's reverbing back through your microphone.
I'm sorry. Yeah, maybe. So should I try to correct that? Or
perhaps maybe when we've got another question here, and perhaps maybe when you're speaking, if you could turn down the volume, I would imagine on your computer, that would probably correct the issue, I assume. But while you still have the volume up, we do have another question, and it's a question concerning what type of instruments, what type of mass specs you used for the work here. Perhaps the question is, could inexpensive, quote unquote, inexpensive mass spectrometers be used? So what type of instruments are you using, and what's your thoughts on utilizing lower resolution type instruments for these types of experiments?
Was my voice bad now?
It still is reverbing a bit. I'm not sure what changed because it was perfect throughout the talk.
Good. This happens to me periodically, and I've never understood it. So usually I just check out and then check back in, and it goes away. So we're using Q exactives for most of this. So, a medium performance instrument, still with good high resolving power. Is that clear what I'm saying?
Yeah, we can hear you. It's a little bit of reverb, but hopefully others, at least I can hear you. Hopefully others are able to make it out as well.
So I think a Q-top, we like the QEs because we get really good MSMS. But once you diagnose the system, once you know where the reactions are occurring, if you're doing a systematic study, their information really comes from an extracted ion chromatogram. So you could use a Q-top instrument for that. So it demands some good chromatography. And by the way, once you add these reagents to the peptides, the retention times change. So the modified peptides elute differently than the unmodified ones, which is really nice. So I would say you could use a Q-top. I would prefer to use a Q exactive. We're doing a very complicated protein complex right now. That's 1.2 million. We're borrowing Illumis to do that. That's because, we have really high dynamic range and very complicated mixtures. That's not necessary. I think with a Q-top or a QE, my answer clear on that?
Yes, I think so. Does anyone else have, hopefully that answered your question there, Stephen. Does anyone else have any other questions? You can either feel free to unmute yourselves and ask away or send it in the chat box. All right. Well, I guess I guess one more ask. Does anyone else have any other questions? If not, I think we're going to thank Professor Gross here for one more question popped in. perhaps you spoke to this a little bit, in that the data is a little bit complicated to analyze. But Mike, how complicated is the data analysis?
Yeah, it's not trivial. When we first started this 15 years ago, it was really messy because we had no software. Protein metrics has helped us a lot by doing some software engineering that allows us to follow modified proteins. So I've put in a little pitch for them. I hope that's I'm not too blatant an advertiser there, but that's helped a lot. It used to be that, you know, we did 1 day of experiments and 6 months worth of data analysis, and it's much better now. It's only 1 month of data analysis. So that's one of the problems. Yeah, now the data analysis is complex, and but it's getting better.
And I know that you mentioned that Protein Metrics is working on this. Do you see in the future that things are going to, I guess they could integrate some of these software solutions into perhaps their software or other software packages to make this a bit simpler in the days to come?
Yeah, I really look forward to that. I think it's really important for this idea to be disseminated. I think the ideas really have a lot of power, and to make them available. So I'm also impressed with Gen Next idea to make a very simple FPOP apparatus with a discharge lamp instead of a laser and built-in optics. So, you know, for safety issues, for laboratory simplicity. Of course, our motivation is to stick with the laser because, you know, it allows us to do these interesting experiments like protein folding.
All right. Does anyone else have any other questions?
Yeah, now that the voice is a little bit better, I had a question which I want to ask. How does the site-specific information, how do you ascertain that? Are you looking at the fragments to localize these modifications? And how is the quantitation possible at a residue level? I mean,
yeah, that's really good.
Specific enzymes or
right.
or do multiple digests, or how do you actually narrow it down to residue level resolution?
So multiple enzymes, multiple proteolysis is a good idea, and we do that. But most of the information I showed you tonight in the chromatography, we'll see a peptide with +16, and we'll see it appear 3 or 4 times in the chromatogram. So what each one of those appearances represent is that peptide modified, say, on residue 2, on residue 5, on residue 6, on residue 8. So they're all isomers, huh? But the nice thing is, with good HPLC, they separate. So the first goal then is to figure out where it's modified. So we do that by MSMS. So we can say, oh, this is modification at point number 6. So in that experiment on amyloid proteins, where I said it was phenylalanine 19 or 20, the MSMS was ambiguous. We knew it was phenylalanine 19 or 20 or both, but the chromatography was insufficient to separate them. But many times the chromatography is sufficient. So now when you want to go back and do an experiment, say in a titration format, for example, or a protein folding experiment, you simply use the chromatogram. So once you've identified it, you know, to be site 6 modified, then we simply integrate the extracted ion chromatogram. So the nice thing is you can imagine that the extracted chromatogram might have, almost never has 6 peaks perfectly separated, but you it'll have some peaks perfectly separated from others, and it'll have some that are convoluted. But you know what they are. You know that this is phenylalanine modification, whereas the other one was tryptophan modification. They elute differently. So better chromatography will always help. And so now to get the percent modification, like I showed you in those residue-specific analysis with amyloids or with protein folding, you simply follow the extracted ion chromatogram as a function of, say, concentration, or as a function of time, as a function of something. So you can just integrate that and compare that to the integral of all of the modifications and the integral to the unmodified. So you put, say, in the denominator, you put all of the modified peptides plus the unmodified, and in the numerator, you put the integrated area of the peak that represents phenylalanine 23, say.
Yeah. So I think, you know, we in pharmaceuticals also do this kind of integration to report modification levels, but, you know, sometimes, you know, ionization really affects how you quantify. At least even for relative quant, you probably get some biases,
Right.
some bias. So with your types of modifications like hydroxyls and, you know, carbenes and so forth, do you expect such a big impact in the modified version to ionize differently to the unmodified, or do you expect them to, you know, be biased in some ways for a quantification?
No, I think it'll be biased. I think you're right. If I think that's what you're implying, that it could be biased. And I think that what you're doing, what saves us in a way is the fact that we're doing a differential measurement. So we're taking the protein folded versus the protein unfolded. And then we're just taking the difference. And so the hope is that the biases will cancel out when you take the quotient. You know, so, okay, we know that this peptide is not ionized as well as its first cousin or second cousin.
Mm-hmm.
So we know that, yeah, so we accept that. So I think ultimately you'd like not to be required to do that, but I think for the near future, we have to do that. We have to, we always have to do it in a differential way. We have to compare the protein with the protein mutant, the protein and the stressed protein. One of them is a control, the other is a test. The protein that's bound, the protein that's unbound. The protein that's at pH 6 with the protein at pH 7.
Yeah.
So yeah, all those peptides, you know, I think you're absolutely right. They will show discrimination. I'm sure you know better than I do, in fact. But when you take a quotient, then of course you get rid of the dependence on ionization efficiency, because both of them are subject to the same experiment. So it's very important to do a very careful differential experiment. Now, it's a good question, but right, we worry about that, but that's our rationalization.
Thank you.
Great. I guess if there are no more questions, Mike, we'd like to thank you again for taking all this time to explain some of this quite interesting work that you've been you and your lab have been working on. You know, we really appreciate your time to present to the group, and yeah, we thank you for your time. We really appreciate it.
Well, and I'm sorry that we can't have a dinner together, but maybe the next one.
That's right. Hopefully ASMS perhaps next year will be in person and we can catch up then.
Okay. Thank you all for listening, and I appreciate your attention. Thanks.
All right. Thanks a lot. Take care.
Yeah. Bye-bye.
Bye-bye.
Bye, everyone.